This video is a recording of the 6th webinar in the SGD webinar series, titled Homology and Disease Data at SGD. Please enjoy.
Hello and welcome everybody. My name is Kevin McPherson. I'm an assistant biocurator for the Saccharomyces genome database, SGD. And in today's webinar, I'll be talking about homology and disease data at SGD and where to find it. Before we begin, if you have any questions, please feel free to ask them at any point in the webinar by using the Q&A button on the right-hand side of the BlueJeans page, as indicated by the red arrow. We won't be using the Interact Live feature today, but we will also be having a Q&A session at the end. So if you have any more questions, please feel free to stick around for that. So let's get started by quickly going over the kinds of homology and disease data we currently curate. Currently, the areas we're focused on and the information you can expect to find at SGD are yeast-human homologs, disease associations in the human homologs, if any, functional complementation relationships between human and yeast genes, where a human gene rescues a mutation in S cerevisiae or vice versa, and lastly, alleles and phenotype variants. If a mutant yeast allele or phenotype is related to a disease-causing mutation or a disease phenotype in the human homolog, we indicate this in our phenotype summary pages. Homology and disease is a currently expanding area of curation at SGD, so keep an eye out as we add more data types and features related to this topic in the future. So how do we find this data? There are three main places you can go. 1, SGD's main search tool. 2, our downloads page. And 3, our advanced search tool, YeastMine. We'll go through these one by one, starting with SGD's search. Running a search is the best way to discover anything related to homology and disease at SGD. To get started, just open up the SGD homepage at yeastgenome.org. Search terms can be as general or specific as you'd like. Let me start with a really broad homology and disease keyword like syndrome. And on the left side of the page, I'll narrow my search results with the genes category, since I'm looking for yeast-human homologs. The genes category will pull up your search terms from all of our gene summaries and descriptions, which you can browse through and see what kind of info is available. If you've ever enjoyed browsing through Wikipedia, for example, where you find all these interesting pages and you just want to keep reading more and more and more, you might find yourself in a similar situation at SGD. For example, HTP1 is shown to have a human homolog, HPRT1, implicated in 2 diseases, Lesch-Nyhan and Kelly-Siegmiller syndromes. Browsing through the page, we can see that TIM8 has a human homolog implicated in deafness dystonia optic neuropathy, and that this homolog can functionally complement TIM8 null mutations in yeast. When you find a gene of interest, click on the link to be taken to its respective locus summary page. Here, you'll see the full description. NFU1 has a human homolog of the same name implicated in multiple mitochondria dysfunction syndrome. Many genes also have a summary paragraph, which can be found by clicking on the summary paragraph link or scrolling down the page. These summaries often include additional homology and disease information. For mutant yeast alleles and phenotypes related to human disease, scroll up to the top of the locus summary page and click on the Phenotype tab. If you scroll down the phenotype page, you'll see the phenotype annotation table, which has all of SGD's curated alleles and phenotype variants for a yeast gene, in this case, NFU1. You can filter the table of phenotype annotations to those related to human disease by searching for human in the filter box. The observable phenotype is indicated on the left side of the table, while the allele or mutation causing it is indicated here. Here we see that these 3 phenotypes are caused by the same mutation. Clicking on the information icon shows that a synonymous mutation in human NFU1 causes multiple mitochondria dysfunction syndrome. Try lots of different searches at SGD and see what comes up. Here are some of the examples of the homology and disease search terms you could try. You can search for something as specific as amyotrophic lateral sclerosis or as broad as disease. Either way, you're likely to discover something new about the yeast genome. To recap, run a search, explore your results, and open up a gene summary page that looks interesting. You can find homology and disease info in the gene description and the phenotype annotation table. Now, there are other ways to search through SGD's gene descriptions and phenotypes for homology and disease info. You can actually download all of our phenotype annotations and gene descriptions as a tab file and search through them with your preferred software. To do so, open up the SGD homepage at yeastgenome.org, click on the download link in the upper right-hand corner of the page, and then click on Curated Data. All of SGD's phenotype annotations are contained within this file. Just download the file. And to find phenotypes and alleles related to human disease in this file, open it up in your preferred program, such as Excel, and search the file for human, like we did in the phenotype table earlier. Also on our Downloads page is our curated list of functional complementation relationships between human genes and yeast genes from the published literature, which you can download and search through just like we did with the phenotype file. Lastly, if you scroll down the page to the chromosomal features section, check out the SGD features file. This contains the entire list of SGD's chromosomal features and coordinates, and includes the gene descriptions we saw earlier when we ran a search for syndrome. You can search the gene descriptions in this file for keywords like syndrome, human, disease, etc., just like running a search on the SGD website. So to recap, our Downloads page is another place you can go to find homology and disease information. Just look through the Phenotype Data tab file for disease-related mutant alleles and phenotypes, the SGD Features tab file for gene descriptions, and the Functional Complementation tab file for functional complementation relationships between human and yeast genes. Now, the data in these three files, gene descriptions, phenotypes, and functional complementation, are all available in one more place, YeastMine. YeastMine is a data search and retrieval tool that enables you to query for, save, and analyze just about any kind of data at SGD. Let's quickly go over how to use it to find homology and disease information. You can access YeastMine by clicking on the YeastMine link in the upper right-hand corner of the SGD homepage, or through the URL, yeastmine.yeastgenome.org. The YeastMine homepage, look for the template queries in the center of the page. Click on the Homology tab to find template queries related to homology and disease, and check out each of the templates available in the Homology tab to see what kind of data they retrieve. Here I'll pick yeast gene to omum human homolog and omum disease phenotype. This query will retrieve the human homologs of any yeast genes you input, along with their associated diseases, as annotated by OMIM, or the Online Mendelian Inheritance in Man database. For example, if I wanted to find the human homologs of yeast rad53, I'd enter this gene in the text field, and then click Show Results. This produces a results table, which isn't nearly as complicated as it looks. On the left side of the table are the genes we inputted, in this case, a single gene, rad53. In the middle are each of the Homo sapien homologs of rad53. The data source of these relationships are indicated on the farthest right column. Right next to the data source column is the diseases column, which indicates if there are any diseases annotated to the human gene by OMIM. If you want to save your list of homologs, click on the Save as List button. Find the portion of the table containing human homologs, and give your list a name. You can access your new list by clicking on the MyMine link in the purple toolbar, and then by clicking on your new list. This will produce a list analysis, which contains all of the genes that you just saved. As a side note, although we ran only one gene, yeast rad53, through the homology query, you can actually query multiple genes at once. For example, instead of inputting a single gene into a query, I could input this entire list of human genes instead. This gets especially helpful if you want to find the homologs of hundreds of genes at once. So to conclude this webinar, I'd like to give one more tip about homology and disease. Part of the essence of SGD is coming in to get connected to data and make discoveries. As a student of science myself, I'm constantly investigating SGD for interesting tidbits of data and places for new research. One way I like to do this is by using the immense data available on yeast genes to predict disease associations in their human homologs. I'll start with a biological mechanism implicated in disease, find all the yeast genes related to that biological mechanism, and then use YeastMine to find their human homologs. Let me demonstrate by using Parkinson's disease as an example. Various mechanisms of mitochondrial dysfunction have been implicated in Parkinson's disease and other neurodegenerative disorders. For example, defects in respiratory chain function, mitochondrial quality control, and mitochondrial organization have all been linked to the pathogenesis of Parkinson's disease. So in this research scenario, I'm going to use SGD search to find all of the yeast genes involved in organizing the mitochondrion, import them into YeastMine, and find their human homologs. To do so, I'll start by running a search for mitochondrion organization at SGD. Note that the first result that came up is a gene ontology term for mitochondrion organization. Gene ontology terms are standardized phrases that describe molecular functions, cellular components, or biological processes, like the term highlighted here. If I click on this link, I can see all of the genes annotated to mitochondrion organization in SGD. But for the sake of demonstration, I'm going to instead find these genes through the genes category. So here we have over 2,000 results for mitochondrion organization. I'll narrow my search results by using the facets on the left side of the page here. Scrolling down, I'll select the gene ontology term mitochondrion organization, direct, to filter for genes that are directly annotated to this term and not related child terms. Click on the term, and then select any other gene ontology terms that can narrow your search results as you see fit. I selected the cellular component term mitochondrion because I only want proteins that are localized to this organelle. Now we have 31 genes that are annotated to the facets I selected. To import these 31 genes into YeastMine, click on Wrapped, and then on Analyze. This will take you to a page where you can import the genes that you just ran through the search into 4 tools. To import them into YeastMine, select the link on the right side of the page. So here is our imported list of genes. YeastMine automatically assigned the name link_1 to this list, and you can change that if you'd like using MyMine. To find the human homologs of all 31 genes in this list, return to the YeastMine homepage and scroll down to the center of the page to find the template queries. Just like we did earlier, select the Homology tab, and then choose the template query yeast gene 2 omum human homolog and omum disease phenotype. Last time we entered a single gene into this query, but now we're going to enter in our imported list by clicking on the Constrain checkbox. Just select the list that you imported in the drop-down menu, and then click on Show Results. And that's all there is to it. So here's an example of one of the results in our query. VPS13 is one of the S. cerevisiae genes we pulled from our mitochondrion organization search on the SGD website, while VPS13C is its Homo sapiens homolog. Since VPS13 is involved in mitochondrion organization, and since VPS13 and VPS13C are homologs, is VPS13C linked to neurodegeneration or mitochondrion organization as well? In fact, just this year, in 2016, a paper by Lesage et al. Came out providing evidence that the human gene, VPS13C, is involved in mitochondrion organization, and that loss of this gene's function may cause an autosomal recessive early onset form of Parkinson's disease. This is just one example of all the different diseases you can investigate, so I hope you'll try lots of different searches at SGD after today. So this concludes our webinar. Thank you for coming, and please keep an eye out for our follow-up email for more information about our next webinar, a 1-minute follow-up poll, and a link to the SGD YouTube channel, where today's webinar will be uploaded.
Thank you for watching the SGD webinar series. If you have any questions or comments, please contact us at sgd-helpdesk@lists.stanford.edu.